When a bacteria containing these plasmids dies, it breaks open and liberates these plasmids to the outside environment, and they are often.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by
The engineers then place the gene segment they wish to insert into the solution with the plasmids, and then add repair enzymes that join the pieces of DNA together at their sticky ends. These new plasmids, with their newly inserted genes, are then placed with bacteria into a different solution that enables the plasmids to readily penetrate through the cell wall and membrane of bacteria. The bacteria that are exposed to the plasmids are penicillin and amoxicillin. They kill off the bacteria that didn't absorb the new plasmids, while those bacteria that did absorb the new plasmids containing the genes for antibiotic resistance and for, say, insulin or growth hormone continue to grow and thrive in the culture. Scientists continue to grow these bacteria until the insulin or growth hormone produced by them is sufficient to be extracted and purified for use by human patients. But how are genes